Hi, I'm Elly De Vlieghere. I'm a molecular biologist at the Laboratory for Medicine in Sint-Jan, Brugge, and I would like to talk to you how we implemented OGM for multiple myeloma in our lab. Now, first, I want to take you to the journey that OGM did in our lab. We started at 2022 when the first Sapphire arrived, and we started with the validation of AML and ALL samples, and based on this data, we received accreditation as the second lab in Belgium to perform OGM. Since then, we have analyzed over 200 AML and ALL samples. Also, in 2025, a second device arrived. And then we started tweaking the DNA isolation protocol a bit so that we could handle low input samples. This with the idea that it would easily make it easier to validate our multiple myeloma samples. And I'm happy to announce that since the beginning of this month, we can actually offer OGM analysis for multiple myeloma in our lab. As I mentioned, we tweaked the DNA isolation protocol a bit, and we actually published this in Method X. This is a journal that describes protocols in detail, so you can actually find exactly how we perform DNA isolation for low input samples. Now, basically, it's just a small adaptation when we are faced with a low input sample, so we don't have the 1.5 million cells available. We adapt the elution buffer according to the number of cells that we do have. And to be able to work with small volumes of elution buffer, we have replaced the tube with a well plate so that you can see here we can elute in smaller volumes. Now we validated this in a range of starting cell numbers from the regular 1.5 million all the way to 500,000 cells. We did this on different samples, so we have bone marrow samples, cell suspensions from biopsies or tissues, and then we have the CD138 selected cells for multiple myeloma. You can see here that actually the quality here of the run here in effective coverage is not changed if we stay above 500,000 cells. So we have the same quality of run. Below that number, we're still able to have a successful run, but you see that our success rates start to drop. Now, it's not only the quality of the run that matters, but also the results should be of the same quality. So what we did here is we took from one sample, it's a bone marrow sample with indication ALL, and we have a range from the normal 1.5 million cells as low as the 500,000 cells. And we looked at the results of the OGM analysis, where we found one balanced translocation with a VAF of 0.45, and we found a deletion of IKZF1 with a VAF of 0.1. And you can see here that actually the VAF is not influenced by the cell number that we start with, so that the adaptation that we did for the isolation does not influence our results or the VAF. With this in mind, we could start our multiple myeloma validation. And what we did is we did a blind study where all the samples that arrived at the lab were parallel analyzed with standard of care and OGM. Standard of care in our lab is FISH and a CNV-Analysis with NGS. Only when both results of all the samples were known, we started the comparison. So first we start with FISH. You see here the translocations that during our standard of care we try to detect by FISH. And you can see that for each translocation that is detected with FISH, we can also detect it with OGM and vice versa. So this results in 100% sensitivity and 100% specificity. You can see here that our blind study, we didn't have a sample with IGH MAF fusion gene. So we did retrospectively, we looked for a sample that did have this translocation, and then we saw that also OGM detects this one. So we can conclude that we can detect every translocation that we are looking for with our standard of care, also with OGM. For the CNV analysis with shallow, we also look for some specific changes in the genome, but we look, of course, at the whole genome with the shallow CNV analysis with NGS. Here we saw in 2 samples that there actually is a difference between our results at the standard of care and the OGM. And when we closely look, we actually saw that our CNV analysis with NGS struggles to detect the correct baseline in complex karyotypes. So what do I mean with this? So this is the results of our CNV analysis with NGS. And you can see here that after analysis, these are the chromosomes that were reported as having 2 copies. These are the chromosomes that were reported as having only 1 copy. And we can already see here some indication that it might be wrong, as this is a male patient and the X chromosome is lower than the chromosomes that are indicated as only having 1 copy. Taking in mind that these are selected cells, so these are pure samples. If you look at OGM, you can see that actually we see the same pattern, but everything is shifted a bit. So where these have for NGS 2 copies, OGM says we have 3 copies. Where there was only 1 copy, OGM says these have 2 copies, and then the chromosome that were below the 1 copy, these are where we only have 1 copy. Together with actually information on loss of heterozygosity and the VAF, we can with confidence conclude that OGM is here correct and outperforms CNV analysis with NGS if it concerns very complex karyotypes. Now, of course, with OGM, we all know we can detect much more than what we just are looking for. And this is one of the phenomena that we also detect is chromotripsis. And we found this in around 10% of our samples. So what is it? It's actually a lot of translocations in one region. Here in this sample, it was in 17q. And this resulted in a lot of copy number variation as well in the same region. A second phenomenon is uniparental disomy, and we actually found it in about 40% of our samples. So how can we recognize this? You can see here a sample with, again, a quite complex karyotype, and we see different regions with loss of heterozygosity. Now we see here chromosome X 22 and 13, we only found 1 copy, and this logically corresponds with loss of heterozygosity regions. But we see also 2 chromosomes that lost their heterozygosity, but these correspond with 2 copies. This means that in this sample, 2 copies of 4 and 12 are present, but they are originating from 1 parental chromosome. And this is indicated as UPD or uniparental disomy. And this is, of course, something we don't detect with our standard of care. So what holds the future for us concerning OGM? So we're going to keep analyzing multiple myeloma, AML, and ALL samples, of course, but we also want to increase our expertise on single cell suspension samples coming from tissue and biopsies. I want to expand our indication portfolio with lymphomas and high eosinophilic syndrome samples. I need to thank the Scientific Foundation for Hematology who funded this work. Also, this work is done by the OGM team at Sint-Jan, but I really want to thank the whole molecular diagnostic team, and I hope you have some questions for me in the Q&A.